This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 257, recorded on December 16, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, Vincent, Michael, all our listeners.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. How is everyone today?
Very well, thank you. One can always complain, but one should look on the bright side, right?
That's right.
Yep. And we're here to provide your twice-a-month fix of microbiology. I want to tell you, though, first, throughout December and January, if you go over to parasiteswithoutborders.com, that is the nonprofit run by my colleague Daniel Griffin, and you make a donation, they will double it, they will match it, and give it back to Microbe TV, the organization that runs TWiM and TWiV and other fine podcasts. So if you'd like to donate, you go there. It's a tax-deductible donation in the U.S., of course, and we would really appreciate that. Or you could donate to Microbe TV, microbe.tv/contribute. We are now a 501c3.
Great work.
Your donations to us. In fact, apparently, it's for all of this year, because as soon as you get the 501c, it goes back to when you first incorporated.
Ah, retroactive. Nice.
Yes. So those of you who have donated, much appreciated, and your donations may reduce your tax burden, which is always a good thing. All right. Now, we have two papers as usual, and I am going to kick it off with a snippet, a cell host and microbe paper entitled Phages encode phage defense systems with cognate self-immunity. And this comes from, let's see, I will tell all the authors to you: Owen, Wenner, Dullberger, Rodwell, Bowers, Barnard, Quinones, Olvera, Rigdon, Rubin, Garner, Baim, and Hinton. From Harvard Medical School, University of Liverpool, University of Basel, Harvard School of Public Health. And do any of you know any of these authors?
Eric Rubin.
You know Eric Rubin? Okay.
Yeah.
Anyway, this is a virus, the second virus paper in a row here on TWiM, which is okay. These are viruses that infect bacteria. And the story here is about the battle, as they say, the eternal battle between bacteria and their viruses, which has driven, of course, defense systems on both sides. Defense systems in bacteria, which include CRISPR-Cas and restriction modification systems, which were discovered long before CRISPR-Cas. Mm-hmm. And then there are some systems where phage infection is prevented at the level of the population by individual cells dying. It's kind of like apoptosis. In eukaryotic cells. When the cell is stressed, it kills itself, and if it's virus infected, that may save the population. Kind of an altruism, the likes of which is difficult to find in humans. I guess I'm being too cynical, right?
You're being a little cynical.
That's my fault for getting you going.
These population-level defenses, they're called abortive infections. And the idea is they prevent phage replication and death. Now, what this paper deals with is what they call a subplot. And that is that many phages, they don't just go in and kill the cell. They don't do lytic all the time. They have there are tempered phages that can integrate their genome into the host and be quiescent for a while. And so then they are prophages. And you know, they have to periodically reactivate and make more phages and transmit to new hosts. And so how did they avoid other phages interfering with that? So in other words, the prophage, the idea here is that the prophage somehow encodes resistance to infection of that same cell with another phage, which may not be beneficial for the prophage. And they say that recent work has suggested that prophage accessory genes are an unexplored reservoir of phage defense systems.
Makes sense.
Mm-hmm.
Yes, it does. And, folks, I am always amazed at what you can always do more with phages and bacteria.
And they're small and yet rich. Yeah.
And you find new things, and they're so manipulable.
Yeah.
It's beautiful. You can do all kinds of experiments, as we see here. So this paper works with Salmonella enterica, subspecies enterica cerovi typhimurium, hereafter S. typhimurium, which is much easier.
Mm-hmm.
It has a prophage. It's about a 40 kb prophage called BTP1. And there is an operon within it called GTR that confers resistance against phage P22. And this is so beautiful. The product of the locus modifies lipopolysaccharides, which is the receptor for the phage P22.
So it
Isn't that incredible?
basically puts up a do not enter sign on the outside of the cell.
Yep.
And once that operon is expressed, it puts up a sign, no vacancy, don't bother me,
It's
leave me alone.
Occupied.
Yes.
Occupied. Now, if you delete the prophage from S. typhimurium, now that bacterium can be infected with P22, right? So it confirms that resistance is conferred by BTP1. However, if you just inactivate the GTR locus, which is the part that confers resistance to P22 infection, it doesn't restore sensitivity to the same level as deleting the whole BTP locus, the prophage itself, right? And so they said, well, this suggested to us there must be a second resistance system encoded in this prophage BTP1. And so they went about looking for it. So they had previously looked at the transcripts produced in bacteria having this prophage, and they identified a gene called BSTA, which they said was a candidate gene for doing such a thing. And when they removed BSTA from the prophage, it increases susceptibility to P22.
Bingo.
And they also put 2 stop codons in and show the same thing. So that it doesn't make a protein now, and now it can be infected with P22. And in fact, if you delete BSTA and GTR together, you now have bacteria that are just as susceptible to P22 as when you remove the entire prophage. So bingo, there it is. 2 loci that are needed for phage defense. And they spend this paper trying to figure out how that works. So the other thing they do, which is really interesting, is they produce this BSTA protein and show that it confers resistance to several phages, not just P22. So it has a broader effect than just that single bacteriophage.
Do those other phages, Vincent, do you happen to know, do they also use LPS as a receptor?
I do not know. In fact, these are phages I don't know of, ES18 and N9A.
Yeah, I hadn't heard of those phages before. And, one of the hallmarks of LPS is the fact that its side chains are all different amongst different. And remember, with Salmonella, there are many, many species that they have since tried to clean up by calling them Salmonella enterica subspecies typhimurium or whatever the proper nomenclature is. And my gut feeling is that they're all just a little bit different in terms of their LPS with the side chain substitution. So I'm wondering if this is a ubiquitous mechanism that Salmonella, the genus, developed to maintain its genetic stability so that all these other phages wouldn't be able to come in. Because prophages are a good way to increase genetic diversity amongst bacteria.
So the prophages triggered diversification into all these different substrains, subspecies. Yep. Cool.
By excluding other phages from invading.
So then they have an experiment that we will actually revisit in a bit. Even if they induce this BSTA that they've put in, and it eliminates plaque formation, when you use a lot of phage, you still get clearing of the bacteria lawn, which they say is consistent with some kind of abortive mechanism. In other words, they think the phage is getting in and it's stopping at some subsequent step. And we're going to come back to that when we do single cell infections in a moment. So these BSTAs are present in diverse gram-negative bacteria. They can just look at the sequences. They find 72 homologs among these bacteria. Most of them are in the chromosomes. Most of them are, some of them are plasmid encoded. So they conclude that this BSTA protein is actually highly associated with prophages of gram-negative bacteria. The protein itself has sequence motifs that suggest it's a DNA binding protein, and that we'll come back to in a bit. So they also took BSTA homologs from Klebsiella and E. coli, and they produced them in Salmonella. And in fact, producing the Klebsiella and the E. coli in Salmonella confers resistance to Salmonella phages at similar levels to the P22, even though the proteins are not very similar whatsoever. They also tested the ability of BSTA to inhibit phages that I would recognize, phage lambda, Phe80, P1, and T7. And in fact, it does, BST from Salmonella does inhibit those, but not T4 and T5, interestingly.
Well, they use different receptors, most likely.
And BST from E. coli is less active against coliphages than the Salmonella BST protein, interestingly. All right, so then they say, let's look at the mechanism. And they figure out that this is working at a population level through abortive infection, which is what they had speculated individually. If they infect hosts at a high multiplicity of infection, so all the cells are infected, they find all the cells are lice, independent of whether or not you have BSTA within 3 hours. So they say it doesn't confer any protection on individual cells. But if you look at single cells, they have a very nice fluorescent assay for looking at single cell infections. If you do a lower multiplicity where you have single cells infected, and then you can see that few infectious particles are produced after infection of those single cells. And so the cell dies, that infected cell will die, but it doesn't produce infectious viruses. So that's the system. It's a population level system that kills cells to prevent virus spread in the population.
So you constrain the phage replication just to that one host cell.
Right. And then you can say, do infectious phages come out of that one host cell? And the answer is no. Right. And those cells then die. They fuse the protein to green fluorescent protein so they can do localization. And they observe that the protein seems to be interacting with the DNA of infecting phages. So it's in the center of the cell, and when a phage infects the cell, this protein is interacting with it. And so they think, and they actually show that this protein inhibits phage DNA replication, which is a bit in the reproduction cycle, right? It's beyond attachment and entry and the early events, the early mRNA synthesis and so forth.
I'll bet that was a happy day in the lab.
I bet.
The protein works right on the phage DNA. That's like, okay, now I can really focus my attention.
Also, you can publish a paper. You have a mechanism, right?
Yeah.
Because if you didn't,
more
we would probably say,
get a grant.
Come back to us when you have a mechanism, right?
Yeah.
This is now, if you're thinking about it, I hope you are. This is a little bit of a conundrum, right? Because the prophage encodes this BTP1 that prevents infection. But what if it wants to reproduce, right? How does the prophage reproduce in the presence of this protein that can stop its own DNA synthesis? And so they say, well, there must be an immunity factor for the phage, right? Something that counters BST1. How do you prevent BST1 from interfering with you when you're trying to get going from a prophage. So what they find is that if you delete the BSTA coding sequence from the prophage, and then indeed the phage cannot replicate because they inhibit themselves. And they actually introduce a double stop codon into the beginning of the coding region, and that did not impair the self-immunity. This self-immunity conferred by this locus. So they concluded it must not be a protein, but something else within the locus. It's not the BSTA protein, which they've shown, co-localizes with DNA synthesis. So something else within this prophage is allowing the phage to reproduce in the same cell, in the lysogen, for example. So they go through some molecular biology, and they narrow it down to a 63 base pair sequence, which they call ABA for anti-BSA. And all they need to do is give that 63 base pair sequence on a plasmid, and it will allow P22 to reproduce in the presence of BSTA. So isn't that amazing?
It's a beautiful result.
You can just figure it all out here. You don't get stuck or stymied like you often do in eukaryotic cells, for example, right? And this does not encode a peptide. It is independent of translation. In fact, it's independent of RNA. It's not based on transcription, but it seems to be this DNA that is somehow the immunity protein to the antiviral BSTA. So if you put this ABA sequence in P22, for example, it increases its ability to reproduce in the presence of BSTA. So it has all the functional activities that you would expect.
They created a gain of function mutation in P22, didn't they?
Don't say that out loud, Michelle.
They did. And in fact, that's a good point, because gain of function is done all the time with all kinds of systems. It's not just bad stuff.
Yes.
And I just wish that the press would really do the homework and understand this. And it's not all bad, folks. But then again, they're influenced by scientists who tell them it's bad, right?
That's right.
The gain of function is bad. And you know who you are, scientists. All right. So the summary here is that you need ABA. Otherwise, the prophage, which has BSTA in it, will inhibit itself by the same mechanism that inhibits exogenous phages from infecting the prophage containing cell, right? They look at these, the effect of these proteins from other bacteria. They find that these BSTA proteins are broadly interchangeable, but each one has a cognate ABA locus, and it's only active against its own. So they're very specific. And then finally, they do some experiments to show that the presence of this BSTA inhibits phage DNA synthesis. And they have some very nice assays, which we won't go into. But if the ABA element is present, then the phage DNA synthesis occurs. If ABA is not present, it's inhibited. Presumably because of maybe the DNA binding part of BSTA is interacting with the viral DNA. So the bottom line here is that this BSTA protein, which is encoded in the prophage in the bacterium, is a suppressor of phage DNA replication. So if any phage tries to get into that lysogen, it's like you're sleeping in a hotel and someone tries to open your door. You got a lock on it, right? Well, the phage has a lock on the bacterial door in the form of the BSTA protein, because when another phage gets in, it's not going to be able to reproduce. And that's conferred by ABA.
So if the hotel gives a new guest a key so that it somehow gets into your room and you're in there, the phage system has a way to prevent the new phage or the interloper from coming in and literally sleeping in your bed.
That's right. That's right. So it's just beautiful, because every experiment is followed through and done in a beautiful way using lovely technology, and they get their answers, which I think I always love when I do phage papers. It's often a consequence. As Michelle said, they're so simple, yet there's so much that can be done.
Well, I was going to wonder if you were going to comment on a couple of their figures that actually have plaque assays in them, Vincent.
They do plaque assays. Yes, they're lovely.
And they actually show you them.
Yeah, it's beautiful.
Yeah, it's very clear.
Lovely figures. Yes, this probably not open access, but beautiful.
No, it
figures.
is. It is, Vincent. It's an open access paper.
How do you get open access? Do you pay for it? Is that how it works?
Yeah, you as the author pays for it. And since these authors were principally from the UK, it was probably the British taxpayer who wrote the check.
Very good. Yes, lovely work. So there you go. These phages have defense against other phages. It's a tough world out there for viruses, right?
Mm-hmm. Well, I think the phage has adapted. Once it found a delightful host to cohabitate with, it said, I'm going to settle in for a while. And it then developed a mechanism to ensure its stability. And if and only if things got really bad, then it would leave.
Yeah. So there you go, folks. Phages have a defense system against other phages, and they have to take care of themselves, so they have a way to counter it.
Mm-hmm.
And this battle has been going on in the environment for eons, yes?
Yeah, sure.
So they developed these really elegant mechanisms that can be described in one beautiful paper.
Yes,
and Michelle's not using hyperbole when she says eons. I mean, it truly is. We're going back a ways.
Well, bacteria have been around the longest, right, on Earth?
Yes.
And so that when they were here, so were their phages.
Yes.
And so there has been a flurry of evolution over the years, and I would have loved to have seen a snapshot of the whole thing. Boy, wouldn't that we get a little bit of a few hundred years here.
Yeah.
But wouldn't it be amazing to see the early days and what happened?
Well, I would posit that the mechanism was probably not as clean as what we're seeing today. I think evolution has cleaned up the mess, which is why it's so elegant.
Yeah, so there was once a phage that infected a bacteria and it killed it. And at some point, a mutant arose that didn't, and there were changes, and those got propagated and selected for. It's all, you could actually sort it out if you wanted to, the way this arose. But
neat biology, Vincent. Thank you.
Yes, it's very cool stuff. Thank you to the authors for doing that. All right, now Michael is going to change to plastic.
There's something completely different. This paper is also open access. It appeared in MBio. It's entitled Plastic-Degrading Potential Across the Global Microbiome Correlates with Recent Pollution Trends. And the authors are Zrymek, Kokina, Jonassen, Zarilla, and Zelniak. And they're from the Department of Biology and Biological Engineering at the University of Technology in Gothenburg, Sweden, the National Institute of Biology in Slovenia, the University of Denmark, and the Toxicology Unit at Cambridge University, and the Institute of Biotechnology and Life Sciences Center in Vilnius, Lithuania. So plastic has become the ubiquitous material from which we manufacture the majority of our enduring goods, from the clothes we wear to the computers we use to, even in some instances, the foods we consume. The opening line of today's paper sets the tone of the challenge facing our globe today. The demands for plastic production are increasing annually, despite plastic waste pollution presenting a major global environmental problem. I personally recycle more than I send to the landfill, but I often wonder where the contents of the blue bin I put out on the curb every other Tuesday truly end up. As evidence, much of the plastic waste does indeed end up in landfills or the great plastic garbage gyres or patches within our oceans. I remember learning of the plastic gyres in our oceans from listening to a TED Talk, and I'll put that link in the show notes for you, from a young man by the name of Boyan Slat. And I was just mesmerized. So it caused me to to look a little further. And the National Geographic actually has a nice story in it, and I'll also put that into the show notes for you to look at. But framing this problem for us as we listen, plastic pieces in the oceans now outnumber sea life by a factor of 6 to 1. That is just absolutely incredible. It's estimated that there are now between 9 to 14 million metric tons of plastic entering the ocean every year, on top of the already accumulated 150 million metric tons of the material that the developed and undeveloped world have deposited into our oceans over the last 50 years.
And if I could just interject,
please.
Next time any one of us is holding a plastic bottle, like the PET bottles, appreciate that bottle, its natural lifespan is somewhere between 16 and 48 years. That's how long it would take to degrade. Decades.
So what this paper is principally asking is, can microbes help address this issue? So let's start with a little bit of vocabulary. First, the good news: some of the thermoplastics, such as polyethylene, which carries the abbreviation PE, polypropylene, the favorite plastic of all molecular microbiologists. It's the stuff that EP tubes are made from. Polyethylene terephthalate, or PET, as Michelle mentioned, or more affectionately referred to as polyester, polyvinyl chloride, and perthalic acid can all be recycled. However, the contaminated and composite plastics, as well as the thermoset plastics, such as the foam in our cushions like polyurethane and vinyl esters, cannot be remolded or heated after their initial forming. So when I was a first-year graduate student taking my first advanced micro class, I learned of the doctrine of microbial infallibility, which was first proposed by Ernest Gale back in 1947 in a book that was entitled The Chemical Activities of Bacteria, where he elegantly argued, and now quoting, somewhere or other some organism exists which can, under suitable conditions, oxidize any substance which is theoretically capable of being oxidized. And of course, plastic polymers are nothing more than a riff on an already pre-existing form of chicken wire chemistry, already pre-formulated by Mother Nature herself as she turned previous biological debris through compression and microbial action into that magic elixir we burn in our cars, namely hydrocarbons. But Gale did not coin the term microbial infallibility. Rather, it was Martin Alexander, one of Vincent's professors from Cornell University, who, as Alexander was trying to refute Gale's concept of microbial infallibility in 1964, Alexander is now credited with immortalizing in the mind of me, the young graduate student in 1981, when I read the following in one of Alexander's science papers entitled Biodegradation of Chemicals of Environmental Concern. And again, quoting Alexander, it is usually regarded as axiomatic in microbial ecology that some organism in nature will utilize as carbon or energy any organic compound. This concept, commonly taken as ecological dogma, may be termed the principle of microbial infallibility. Now, Alexander coined that term back in 1964, and he wrote his science paper in 81 when I read it. And needless to say, I have carried this concept with me until this day and was delighted to see the paper we're going to do today or discuss from MBio specifically address advancing that concept. So today our exploration of this dogmatic concept is critical to the question posed by the authors in this paper, namely as to whether the human race, through our manufacturing and gluttonous consumption and disposal of these complex carbon-based polymers, are trying to circumvent the dominant life force on our planet, namely the microbes. So first, at the outset, the majority of microbiologists will offer that their understanding of microbes capable of degrading plastics is poorly understood. However, the authors here are taking advantage of the advances in the large number of global environmental DNA sampling and sequencing projects, and then through the construction of hidden Markov models from experimentally verified enzymes that have been documented capable of attacking the common forms of plastics, then using that in concert with ocean and soil metagenomes, they're able to ask the fundamental question, or the 150 metric ton question: what is the global potential of microbes to degrade plastics? Now, they segregated their results into two declarative sentences that really gave me a great deal of hope as I, got into this topic of the plastic and the accumulation of plastic in our oceans. That the plastic oceanic garbage gyre could potentially be subject to microbial degradation. The first telling statement was the global microbiome does indeed harbor thousands of potential plastic degrading enzymes. And the second statement that they offer us in presenting the results is that the Earth's microbiome's plastic degrading potential might already be adapting to our global pollution trends. Think about it. In the short 50 years that we have been pitching things over the side, so to speak, the microbes have gone after this very valuable carbon and energy source. Remember, plastics from the microbial perspective is nothing more than a very reduced form of carbon with all sorts of energy. And that Mother Nature has taught the microbes that they can indeed use these compounds because the natural oil seeps in our oceans that Mother Nature occasionally leaks out due to volcanic activity or just leaching, microbes can indeed go after. We learned that in the Gulf oil spill of 2010. So their first result,
and if I could just amplify on that,
please.
the Deepwater Horizon oil spill that Michael just referred to, when scientists went in and studied the water, they found that there were microbes there that bloomed that were able to degrade that complex oil molecule.
And when I say chicken wire chemistry, that is really an homage to one of our TWiM colleagues, Rich Condit. He uses that term when he's describing organic compounds. And if you've ever looked at the structure of plastic, it really does indeed look like chicken wire, except there are various side chains like a chlorine or bromine that they tack on to effectively make it resistant to microbial attack. So anyway, back to the paper at hand. Their first result, that the global microbiome harbors thousands of potential plastic degrading enzymes, was determined as you might have forecasted. First, the authors went to the literature and they compiled a data set of known enzymes, and here's the underscore, with experimental evidence of plastic modifying or degrading activities from published studies. They just didn't simply say, okay, this looks like it'll degrade plastic. They went and looked for the evidence that was either annotated in a database or from a paper itself that presented evidence. Recall that the ideal way to disrupt the integrity of any of these complex chemicals or polymers is to break the structure, either through oxidation or reduction. But the goal is to disrupt the structure. In other words, they went looking for novel wire cutters. So starting with their initial set of 95 experimentally verified and published sequences of plastic degrading enzymes, they then expanded it with this hidden Markov modeling approach using the UniProt database and which has lots of sequences to build their enzyme sequence models. I'll put a paper into the show notes entitled Hidden Markov Models and Their Applications for Biological Sequence Analysis that was published by Yoon in Current Genomics in 2009 that walks the reader through a tutorial of how hidden Markov modeling can be used to solve various sequence analysis problems. The authors in their methods tell us how they did it, but it wasn't enough for me, so I went and did a little hunting, and I found this one paper that I think will help the novice reader get into the value proposition of how hidden Markov models can get things done. To just give you a taste, the hidden Markov model is actually a statistical tool that can be used to describe the evolution of observable events that depend on internal factors which are not often directly observable. So I believe our hidden aspect here is the enzymatic potential. And the hidden states form what is termed a Markov chain, and the probability distribution of the observed symbol in the model depends on the underlying state. And for this reason, the hidden Markov model is also often referred to as a doubly embedded stochastic process. And if you know anything about weather forecasting, this is effectively what the forecasters are using. Now you know why I put that paper into the show notes, and their methods will help you get comfortable with their data.
But in general, Michael, is it an oversimplification to say in general they were looking for sequences that shared a fair amount of DNA identity, sequence identity?
That is a simplification.
Yep.
But so, it is indeed what actually happened.
More sophisticated than that.
It's much more sophisticated. And that's, you know, one of the other reasons I dumped the paper in the show notes is Markov modeling is going to become more and more important as we begin to think about what comes next after COVID and, you know, all the what's next experiments. So results from their exploration is found in their first figure, which principally walks us through how they did their enzymatic mining, because that's effectively what they're doing. They're doing enzymatic mining of the genomic databases. And that's seen in panel B. And they just walk us through this very straightforward approach. And from their exploration, they uncovered 30,000 potentially plastic-degrading enzymes. The first came from ocean databases where they surfaced, I can't resist the pun, 11,906 potential hits from 67 locations sampled at 3 depths across 8 oceans.
And I just have to say, I so admire these this research team. They really were trying to be very comprehensive here. We're very bold in the scope of their study, weren't they?
That is it's saying something.
So when they say global microbiome, they can justify that. They really
well, that's their second figure. They give us a map of the globe and show us where they sampled.
Right.
And so you can actually see the databases, the location of the soil and ocean samples that they actually hunted. Now, for soil microbiomes, they looked at 169 locations from 38 countries from 11 distinct environmental habitats, and they found 18,119 potential hits, which they posit could serve as 10 unique wire cutters for the major types of plastics, the 6 polymers and the 4 common additives. What's even more remarkable, but likely not surprising to anyone who subscribes to the Gale and Alexander's thesis of the dogma of microbial infallibility, when you look at the identity of the enzymes, and this is figure 1, panel D, nearly 60% of the enzymes they identified didn't map to any of the known classes of enzymes that are routinely thought to break chicken wire chemistry. So the oxidoreductases, the hydrolases, the lyases, the transferases, 58% of them were not that. So
and that reflects how young this field is of looking for microbes and enzymes that break down plastics.
And so this also tells us we have to get better at annotating the microbiomes of the organisms that we're sequencing to figure out what these proteins actually do.
I have one word for you, Michael.
What?
Plastics.
Oh, God. I was waiting for when that was coming.
You timed it well. Michael, we're lucky to have these sequence databases, right? People have gone out in the past and sampled soil and oceans. This is the part of the beauty of it, right?
Great resource.
It's something that I don't think Martin Alexander or Gale could have ever anticipated that they could go hunting. So just from their first figure, they offer us that novel plastic degrading functional content can be revealed by their probing strategy. And this, again, should not be surprising considering recent revelations of vast numbers of novel functions uncovered in recent large-scale metagenomic studies. So I guess this is my impassioned plea or asking for a New Year's resolution is if you're part of these metagenomic studies, go the next step and try to document or do some experiments to figure out what some of these proteins are doing. Now, in fairness, this work group was really rigorous in critiquing their own work or their approach to go on this expedition looking for plastic degraders. They offered that they use the gut microbiome as a negative control, since as the premise of their initial hypothesis is that such a microbial community would not have been selected to degrade plastics. After all, the human race has not been eating plastics by choice, thereby allowing them to offer that plastic degrading enzyme hits links to enzymes in our gut would likely be a false positive. So they were using our microbiome data to subtract out any of the hits. Now, the problem with that supposition or hypothesis is that there are data from recent studies offering that the human race may be indeed ingesting large amounts of plastic particles in the form of micro, which is less than 5 millimeters inside plastic, and then micrometer or nanoplastics that are less than a micrometer inside. It seems that our food supply may be bioaccumulating plastics. And in point of fact, foods like carrots, lettuce, apples, pears, all contain this form of plastic. Yes, I went down the rabbit hole and was surprised to learn that apples had one of the highest microplastic counts in fruit, with an average of 195,000 plastic particles per gram.
My gosh.
Michael, were they stuck to the outside of shiny apples?
It's in the inside. While pears averaged around 189,000, but the thing that should give all little kids great solace is broccoli and carrots were shown to be the most contaminated vegetables, averaging more than 100,000 plastic particles per gram.
That's huge.
This was from a Greenpeace post that I found on the net. And, Greenpeace goes on to say how Hong Kong's Consumer Council also said that salt can harbor micro and nanoplastic between 114 to 1,700 milligrams of microplastic per kilogram of salt test.
Michael, in that food paper, did they speculate that it's coming from the water supply?
No. Yes, it's being uptaken by
through the roots.
Through the roots, and the root system has it. And some of the science papers that I then went down the rabbit hole and looked at argue that it's being literally imported into the plant structure.
And how is it getting so small? Is this by physical abrasion in nature of the plastic?
Physical abrasion is what they think is going on, because it's not being degraded to its base blocks.
Yeah.
So
and
all of this
what's the consequence for us, Michael, eating all this?
That's next. All of this plastic in our diet cannot be good for our microbiome. And they offered, but this is another topic for TWiM, but just to close out this thread, they offered that plastic contaminants, according to cited studies of animal models in this particular paper, can result in impairments in oxidative and inflammatory intestinal balance, disruption of epithelial permeability, and of course the big one, dysbiosis, where a microbiome is actually altered, where the symbiosis between host and the natural community is being disrupted.
And just to underscore that this isn't far-fetched, the authors also name a couple species of worms, mealworms, waxworms, that have already been found to have a gut microbiome that have species that can degrade some of these plastics.
Specifically polyethylene and polystyrene, or both plastic types simultaneously.
Wow.
And remember, these worms, like the great waxworm and the waxworm and the mealyworms, which actually will grow up to become moths, actually in their larval state have complex microbial digestive systems that will literally degrade it. If you will, they are likely going to become the termites of the plastic world. Because after all, termites are designed to degrade wood to get their carbon and energy. And wouldn't it be swell if we could exploit the worms and mealyworms to effectively get rid of some of this plastic?
So, Michael, are you going to go through that troubling data where they actually look at different regions that have more or less plastic contamination and then ask how many plastic degrading enzymes did they find?
Yes. So you might be wondering, as Michelle is bringing up, how did they explain the ocean was not as rich in enzymatic hits as the soil they probe? And they offered us that the ocean, given its highly dynamic environment, it's also a little bit more dilute than the soils, where everything is in a non-compositional medium in the ocean, where there's a large degree of mixing, which soils are effectively composed of solids. You can expect an intrinsically lower community and functional stratification in the oceans as opposed to the soils. Their figures, since this is an open access paper, I'll commend you to look at those specific figures. Michelle, that's the extent that I was going to go into it. Do you want to go in a little bit more?
Yeah, go ahead and point out the figure.
It's probably figure 3 and then figure 4 again, where they're measuring the pollution per country and the share of plastics inadequately managed and the number of plastic degrading enzymes. Correct?
Yeah. So the implication is that we have changed the environment and now the microbes are taking advantage of this new opportunity. But it's just really sobering to realize that we have not left no trace.
And in fact, in their discussion, they really drive home the point. They offer us that we have the first signs of global plastic pollution that was raised over a half century ago. And in fact, we now have seen the beginnings of adaptation and the degradation of PET, or the polyethylene terephthalate, or the polyester degrading enzymes in the planktonic communities, where multiple fully functional enzyme variants were found to have evolved from the ancestral enzymes degrading these polycyclic aromatic hydrocarbons. If you will, the original chicken wire that I alluded to in the beginning, that have been seeping into our oceans for millennia, suggesting that current PET exposure already provides sufficiently strong selective pressure to direct the evolution and repurposing of such enzymes. Now, the fundamental challenge here with all of this, it's great that the microbes have the potential, but the problem is speed. There is no doubt that all that carbon can be energy for microbes, but it's a matter of time. And we don't necessarily want to be eating plastic in our apples, pears, carrots, and broccoli. They offer towards the end of their discussion that the predictive lifespan, as Michelle pointed out earlier, that polyester bottle, the water bottle that seems to be ubiquitous at faculty meetings, can last for 16 to 48 years. So we as microbial physiologists need to embrace our colleagues from chemical engineering and synthetic biology to enhance current plastic degradation processes if we are going to cut the amount of plastic that persists in our soils, oceans, and even our foods. They leave us with the following closing argument that warrants repeating, quoting, "Although there is still unexplored diversity in microbial communities, synergistic degradation of plastics by microbes holds great potential to revolutionize the management of global plastic waste." So to end this, the methods and the data they present on novel plastic degrading enzymes should provide us hope, and more importantly, in how critical it is to understand the mechanism and the steps involved in the biological breakdown of plastics, offering then a team science approach from microbiologists, chemical engineers, synthetic biologists, a basis for the development of a viable and cost-effective approach for industrial plastic waste biodegradation. And it was an eye-opener for me. And Michelle, what have you learned?
Well, speaking of interdisciplinary science, and clearly this shows what a demand we have, there is hope because we have talented scientists like our first author, Jan Zermik, who I was able to get a profile on. So he was originally studying electrical engineering in his home country of Slovenia, and he got his PhD also in electrical engineering. But then for postdocs, he did joined a number of different labs and a number of projects studying biotechnology, various synthetic biology projects. And till he joined this group in 2018, went back home to his native Slovenia and was fortunate to join this group with the National Institutes of Biology and got research funding to really apply his expertise in systems biology, deep learning, to come up with this really logical but comprehensive bioinformatic approach to identify potential enzymes. When he's not doing this rigorous work, he appreciates that he has to maintain a balance. So in his spare time, he likes to do Tai Chi and yoga, or unwind with some music, reading science fiction, or meditation in order to fight off the fatigue of the working and the hard thought each day. So he encourages us all to keep our mind and body in good working order. He also was fortunate to work with master student Sarah Johannesson to set up this procedure, and they worked closely, and then also had a couple of visiting researchers and a previous master student, and this is Kokina and Zorilla, who are now doing their PhDs in this field as well. So it was a great team doing this work. And he says a key turning point came when he saw a post about plastic pollution and the global figure of how much plastic sampling had been done. So he knew that they could get their hands on these data sets and use it to develop this really sophisticated probe for new enzymes or unknown enzymes that can degrade plastic. So we need more people in this field because there is a hell of a lot of plastic out there. And I'll just say, I now, when I'm shopping for various products in the grocery store, I'll like go up and down and squeeze all the bottles and find the one that's made of glass, because although we can throw our plastic containers into the recycling bin, there's not a large enough market for it. So a lot of that actually is not being recycled.
Yeah.
It's being put in the garbage. So glass and cardboard, until we can get the microbes working on our behalf.
And aluminum cans.
And
don't
aluminum
forget
cans.
Aluminum cans.
Yep.
And the good news is that all the pasta sauce is in glass pretty much. So for some reason, it never made it to plastic. I don't know why. I guess
But
when you
olive
drop
oil,
it makes a mess. Olive oil can be in tins. But it's often in plastic. Yeah.
But you can find glass. Just squeeze until you find the brand that's in glass.
So, Michael, what about meat, fish, poultry? Does that have plastic particles too?
Oh yeah, I didn't get into the scary part. Ocean fish have plastics in them as well.
Sure,
salmon, carp.
Oh my gosh,
mullet.
All right. So then, what do? You take these sequences, you make the enzymes, and you see if they work. Right? Is that the plan?
Principally, that's
then at
the plan.
some point, you're going to throw bacteria with these enzyme systems in them into the oceans, right?
No, I think that there are plans to go and accumulate the plastics out of these gyres, and actually, once we get a plant set up on land, we'll begin to clear them up, because I think
that's an expensive proposition to harvest and then transport all that plastic.
Well, or you have it done in situ on the ships, where they convert it into energy or something. I mean, that's for a systems engineer to figure out, not me.
It's not straightforward, because if you throw the bacteria in the oceans, they're going to overgrow, right? With
Pretty
all that
much.
plastic, and then you're going to have another problem. So,
well, the population will contract once their job is done, much like
maybe
degrade
you
oil.
need to take the recycling and throw it and bring it to centers, and then there degrade it, right? What's in the ocean may be long gone, but I don't know why we keep throwing stuff in the ocean. We need to stop.
Well, you see what's outside of Staten Island, the Fishkill landfill.
Oh yeah, it's huge.
It's I think the highest point on the eastern seaboard.
Michael, every time I use something, I just think of the billions of other people doing the same thing.
Yes.
And it's multiplied. You know, I put out a bag of trash, and I'm thinking of how many bags someone just put. It's too much.
Yeah.
I don't know how Earth takes it. Earth is very resilient, but it's not going to last forever, right?
So this is a call to all our aspiring scientists out there. We have one word for you, and that's plastics.
Got to figure this out.
and we are quoting from the 1967 film The Graduate, where Dustin Hoffman is given this advice by a family friend, Mr. McGuire.
I think it makes perfect sense. The bacteria can do anything, so let's unleash them onto this. It's a great idea.
And the dogmatic concept that I learned as a tiny graduate student of microbial infallibility is still holding. And I also put into a show notes the paper that was in FEMS, Rethinking Microbial Infallibility in the Metagenomic Era. And it's an interesting read. So if you're interested in doing some over the holidays reading, it's an interesting story to consider.
I think that was great. Thank you, Michael, for bringing up our awareness. I never, it was just good to have. I appreciate it.
Well, you've been dropped into the deep end of COVID for 2+ years, and so
are you implying that I'm not thinking of much else? Because you'd be right.
Well, no, you're just overwhelmed with all things COVID. You've been doing 3 TWiMs a week on nothing but.
Yeah,
it's great to hear you talk about phage on TWiM. You're welcome to talk about environmental phage and bacteriophage.
Yeah, no, I'm happy to do that, and I do look forward to the day when we don't have to talk about COVID all the time. It sounds funny. I mean, I do want people to stop becoming ill, but it's just not right to focus so much on one virus. And, you know, there are interesting things to sort out, but there are other really interesting viruses out there, and I look forward to getting back to it. So,
well, I mean, just think about it from a perspective of climate change. All that carbon is going to be converted ultimately to CO2. And it had been sequestered for millennia. in the form of complex hydrocarbons that were trapped.
Yep.
And we have been pumping oil to make these plastics.
Yeah, we messed the whole thing up. We think we're so bloody smart, right? We messed it all up. All right.
That's TWiM.
That's
time for scientists to get on the case.
That's right.
TWiM 257. You can find show notes at microbe.tv/TWiM. As I indicated earlier, if you'd love to support us, microbe.tv/contribute. Questions and comments, TWiM at microbe.tv. Michelle Swanson's at the University of Michigan. Thanks, Michelle.
Yeah, and I want to thank each of you for another great year of TWiM.
You bet. That's our last episode for 2021.
Yeah.
Yes.
Here's hoping 22 is better.
I do hope so. Yes, good sentiment. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thank you. And I'd like to wish you a premature happy birthday, Vincent.
Thank you.
By the time we do our next TWiM, you'll have gone around the sun one more time.
One more time. You bet. Thank you. I also want to thank Elio Schechter, who's not here today, for all his contributions in the past year, and hope for more from him as well. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.